All right, going off of what we left off from last class, you're going to take back out that packet that had transcription and translation, and we're going to do our translation notes today before you do some practice and you do an exit ticket. If you didn't get the packet from last time, make sure that you grab one from the front or from the sub, and then you're going to go ahead and just start with the transcription part from the previous class that you missed. It's all in Google Classroom, and it's marked by date and by day. So today's learning objective is students will be able to explain the purpose and process of translation using models and recognize how it is a regulated process. In your journals, you're going to write that down, and then you're going to answer the two do not questions. The first one is, why can't the DNA directly make the protein? And the second one is, how are the 4 nitrogen bases of mRNA different from the 4 of DNA? You're going to get that answered in your journal, and then I'm going to check it when I come back. So pause and go ahead and do that. So now that we have sent the instructions with the messenger or the mRNA, it's time to actually get the protein made, which is the whole point of the central dogma. And that's going to be done through translation. And that's going to happen outside of the nucleus, inside the cytoplasm. So after transcription, mRNA leaves the nucleus through nuclear pores or openings or holes in the nuclear membrane, and it'll move to the ribosomes in the cytoplasm. They're the protein factories, so they're going to make our proteins for us based on the information the mRNA is carrying. A 3-nucleotide sequence of mRNA is referred to as a codon. So we talked about all the bases last time, and you learned how to convert the bases, but we group them into groups of 3 at a time in a codon because that's how they're read by the ribosomes and by the tRNA. So it's important to understand that these are specifically referred to by this vocabulary word. So tRNA is called transfer RNA, and it's called that because it transfers the info of a codon to an anticodon and then to an amino acid. So there are molecules that have anticodons on them that are complementary or matching to the codons in mRNA. So there's going to be 3 from the mRNA, and it's going to match with 3 from the tRNA. On the other side of the tRNA is the particular amino acid that the codon codes for. So you have your mRNA codon, you have your tRNA codon, and then at the tRNA anticodon, and at the very opposite end of the tRNA, you're going to have an amino acid that is going to be able to be attached to many other amino acids based on what was brought in from the codon matching with the anticodon. Ribosomes take those amino acids and attach them together, forming a polypeptide, or a peptide if it's short, chain, and that's also known as an a protein. So polypeptide chains will keep growing until that codon from mRNA at the bottom actually signals for a stop instead of signaling for a amino acid to be added. So here we have the anticodon at the bottom of the tRNA, and you can see it's called a tRNA, and it helps remember this because there's actually like a lowercase t shape to it. And on the other side of the anticodon that will match with the mRNA, there's an amino acid here, which is arginine in this case. This anticodon is a triplet as well, and it matches with a triplet of mRNA. That mRNA would have been CGC if we had seen it. There are 20 amino acids total, which means that because there's a lot more codons that exist than amino acids, there's going to be multiple codons that will code for the same amino acid. So some amino acids have 6 different codons that make them, and some only have 2. So some are specific, some are kind of general, and some are more common because of that, and some are more rare to find because of that. The genetic code, you can kind of simplify in your head by thinking that letters are like the 4 bases on mRNA, like A, U, C, and G. And then we can use those letters to make words, which are our codons, the 3 mRNA-based sequences. The example we have here has a mRNA sequence, AAA, CGU, UCG, CCC. And if you can tell from the way that I even read it, I already in my head break it down into the codons, 3 bases at a time. So AAA, CGU, UCG, and CCC are 3 separate codons. And when you use an mRNA chart that I'm going to introduce later, they will be corresponding to different kinds of amino acids that are linked together in this chain. And this chain is only 4 long, so it would be a peptide chain, but that's the protein. Now we started off with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 bases, but in the end we only have 4 amino acids because when you have the nucleotides, it takes 3 of them to make a codon, and 1 codon then makes an amino acid that's coded for. So you're going to be shrinking the numbers a little bit. So as an example, think of 27 bases in your mRNA. You're not going to be making 27 amino acids in that chain. Instead, you would divide it by 3, and you would actually get a 9-chain amino acid of a peptide, right? So then it would be a lot shorter than the initial, 27 bases that you had. Here's the big picture kind of visually shown as well. We've had this top diagram a bunch of times now, but the bottom one shows the DNA matching with the mRNA that's coded off of it. And then you're breaking them into kind of sections of 3 bases at a time to isolate and think about when you're converting it into the amino acids that you're going to link together into the protein chain at the end. But how do we know which amino acid each codon makes? Well, there's a simple way to do that. We have this universal genetic code chart, which we use with mRNA, and specifically only mRNA. There's first base, second base, third base, and we do it similarly like baseball does, where you go from first base to second base to third base inside of your codon because there's 3 letters at a time. And hitting home plate is when you actually find the amino acid that's associated with the codon you were using. So let's do some practice. Here we have TAG as our DNA triplet. And we don't always read DNA as triplets, but in this case, we'll use just 3 at a time because it already matches with what our codon would have and our anticodon would have. So we're going to use the rules AU, GC for mRNA to match with DNA. So T will match with A will match with U, and G will match with C until we get AUC in mRNA. Now, we're going to find out what the anticodon on tRNA is, but in the end, we're still going to use the mRNA to make the amino acid. Okay, so the tRNA anticodon will match with the mRNA codon, which means A will match with U will match with A, and then C will match with G. So we have UAG as our tRNA anticodon. And if you think about it, it's pretty similar to the TAG, except it's UAG because RNAs can't have Ts. So now we're going to take the mRNA codon because this is a mRNA codon chart. Even though we found the tRNA, we cannot use that as a chart because the title of our chart is mRNA. Okay. So we're going to find out the amino acid that the mRNA here actually codes for by using our codon chart. And so we're going to do, like I said, in baseball, we're going to go first base, second base, and third base. Well, the first base here is A. So we're going to look on the column on this side where it says first base, and we're going to find out where A is. And A turns out to be this whole row right here. Perfect. The second base inside of our mRNA codon is U. So where it says second base at the top here, we're going to look for U. And U is this first column here. And where these two actually collide in the middle, we're going to be looking in this box only now. Because inside of this box, they all begin with AU, 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 AU. Perfect. Now we are going to go ahead and do the last one. I still have a typo here. That's my bad. So we have A, U, and C. And C is our third codon base. And our third codon base is going to be C. And we're looking on the third base over here because the right side says third base. So it goes U, C, A, G. And we're looking at only the row where it says C. And to double check, we have AUC here. Perfect. And the arrow goes to ILE. That means that our amino acid is going to be ILE. And that's going to actually stand for isoleucine. So now that we found the amino acid, we figured out this whole row up here. And we figured out actually even what amino acid would be attached based on the DNA from the very start. Now, for the rest of this table, you should be able to go back and forth and change the mRNA into DNA, change the DNA into mRNA, change the mRNA into tRNA, and kind of toggle and see. But the only part that I really want you to focus on is once you get the mRNA, you're going to use this column to find the amino acid at the very end. Okay. I'm going to ask you to pause this video and try the rest. All right, so you should be able to check your answers now that you've tried the rest after pausing. Here you go. Afterwards, you're going to do some more practice, and this practice isn't exactly like the table, right? You're going to just use the codon from mRNA and write the corresponding amino acid. And then the bottom one's kind of fun here. There is an amino acid for alanine, and there's an amino acid for serine. And they each have multiple codons that can create them from the mRNA codon chart. So you're going to do the opposite here. You're going to hunt for the amino acid name and find out which codons can actually create them and list them for me here. Pause and go ahead and do that. Here are the answers if you would like to check it. For serine, there's actually 6 of them. So if you just wrote 2 out of the 6, that's totally okay. So again, transcription is the process of turning DNA code into mRNA code, and it lets the instructions travel from the nucleus where the DNA can't leave and go to the cytoplasm through a message. So when we're going from our DNA, we make our mRNA. Here we would go UCC, GUA, CGC, AAU, and UGU to make our mRNA. But then we would take our translation and we would turn the mRNA code into an amino acid sequence, which is the end goal that we were trying to aim for. Anticodons on the tRNA match up with the codons on the mRNA to bring in the specific amino acids. And the codons are triplets of nitrogenous bases. The anticodons are as well. This will happen in the cytoplasm on the ribosomes. And we have our example of UCC, GUA, CGC, AAU, and UGU. For bonus points, I would love it if you could find the amino acid sequence that is associated with these 5 codons. This is our amino acid chart. You can use this one or you can use the one on your paper to figure that out. So our overview again is you're going from DNA to RNA to proteins, and protein is always the end goal because that's the whole reason what the instructions are for in DNA. Whereas it's happening as going from the nucleus where the DNA is, where the RNA is copied from it, and then the RNA will leave the nucleus and go to the cytoplasm where the proteins will eventually be made. Transcription happens in the nucleus. Here we can see RNA polymerase. And then translation will happen in the cytoplasm where we can see the amino acid chain being made based on the mRNA using the ribosomes. Our end product is our proteins, which build up the physical body of most living organisms. And now you're going to go on to the practice part. We did a little bit of practice learning how to use the codon chart, but you're going to do some more practice now in your independent practice on your interactive notebook for week 20. After you finish that section today, you will actually turn it in through Google Classroom. So make sure you hit that turn in button once you finish. And then you're going to do your exit ticket for translation. It's also in Google Classroom marked by date and day. I won't see you either at the beginning of next week. So have a good weekend and I'll see you soon.